The overall goal of this procedure is to create adhesive and soluble gradients to study cell migration. This is accomplished by first passivating a surface to prevent nonspecific cell adhesion. The second step is to fabricate stamps from microcontact printing. Next, the passivated surface is patterned with streptavidin lines by microcontact printing. Streptavidin can also be printed as dots using dip pen lithography. Attached to the bottom of a microfluidic device, the modified surface will act as a foundation for an adhesive gradient of biotin RGD. The final step is to load cells onto the surface with the adhesive Q in the presence of a soluble chemoattractant gradient. Ultimately, live cell microscopy is used to show cell migration in response to both adhesive and soluble cues.
The main advantage of this technique over existing methods such as transwell or Boyden chamber assay and a conventional microfluidic setup is that this integrated system of surface pattern microfluidic device allows the observation of cells' responses to spatially controlled adhesive cues, an adhesive gradient, and a soluble gradient at the same time.
Well, this method can help answer key questions in the cell migration field, such as the importance